threats. Do you want to sit here, Albert?
Yes, I'll sit here. Thank you. I think we're all very informal here. Thank you very much. I'm greatly appreciative of the opportunity. And as far as I can understand from Johan and Brian's first emails to me, my charge today is to basically update you on what I said about these issues 2 years ago in Estoril, and I shall do so. Who is controlling this machine? Can I have the first slide? This is just a disclaimer to say that I did have someone paying for me to come here, but I want to emphasize that any opinions which I shall share are purely my own and nobody else's, although I live in hope. The next slide, please. So I'm going to cover I'm going to basically share what I think, and I think basically I'm here in a purely personal capacity, not representing anybody really, except myself, which is more than enough. But I will share some perspectives on the issues which we spoke about 2 years ago. I was charged to give a long presentation on this whole issue, which I think will be looked at by other people today. I will say something not just about hepatitis C, but very briefly about emerging viruses. And since it was something which did creep up again on the landscape last year, I thought I would take some time to say something also about TSCs. Next slide. So I think that 2 years ago we had a very vigorous discussion about the whole issue after the aftermath of the relaxation by the FDA, and we looked at various deferrals, and things since then have not really very changed much. This is a survey from Transfusion last year, and basically the point I seek to make from this survey, and I will touch upon this concept in a few minutes, is that a considerable number of individuals who donate blood do so with the knowledge that even in relation to current deferral and restrictive policies, they are basically legally in breach. And this is something to be kept in mind. It's not something which is necessarily of any concern at all to this community. Much of what I'm going to describe is in fact of no concern at all to this community, for the simple reason that plasma-derived medicinal products are very strongly buttressed by a layer upon layer of safety, which make most of these measures very relevant. Can I have the next slide? This is now the topic which I think Johan in particular wanted me to touch upon, and this is the fairly recent evolution of so-called HIV pre-exposure prophylaxis, whereby individuals who are knowing that they are going to enter into a behavioral or some other aspect of exposure which is associated with high risk to HIV, take certain drugs which are now well known in relation to decreasing the risk of infection. And this is, of course, the early initiation of ART therapy. There is a controversy as to what is the actual effectiveness on this. It's remarkable, the spread of effectiveness data which you get from the literature. It is based, this variability, on the fact that even though it is available and is accessed, the actual compliance to the letter of the label of these drugs in relation to what you do is very, very variable. If you do actually fully comply, there should be a very high level of efficiency. There is a possibility that the interference of this therapy should be highest in the context of concomitant low-level positive donations, which give us our historically, traditionally ambivalent results in the serological and NAT assays. And this is something to be kept in mind, that probably with these drugs, this is another factor. And I shall expand upon this in the next slide, please. This is a very interesting cartonic representation from a recent editorial linked to a very important paper to show the shortcomings of our current diagnostics, both in terms of antibody seroreactivity, but also in terms of the antigen NAT detectable aspects of the infectious event in HIV infection. And in regards to this slide, I draw your attention to the contrast between the top and the bottom panels, which show that with the initiation of this kind of therapy, you get a down regulation of the immunological and the virological replication response to levels which are below the limit of sensitivity of different generations and different technologies of the assays, but which can, in fact, result in infection. Incidentally, I want to say that, as usual, in fact, the organizers have a much expanded version of this presentation, which I curtailed because of time limitations, which is available to anybody. I mean, this is all public information. The next slide. A very nice, sexy representation of individuals who are actually studied in the article which the previous slide editorialized, which basically shows the point I have just made, that if you look at the various subjects in relation, each number is a subject, as you can see, and you can see those dots showing you the positive or negative results in the various assays. There are different generations, okay? Different generations of immunoassay as well as Western blots on the right-hand side. And basically what you can see is that you get a positive result being elicited, which then becomes negative because of the ART. So I think that this is of interest and significance. It's probably of interest and significance, as I said, in areas other than the question of the safety of plasma-derived products and the whole issue of MSM deferral, but it has to be kept in mind that this is definitely being found. The next slide, please. I now say something about the evaluation. This is an interesting paper in Transfusion. It was published after the last PLAS meeting, so I thought I would make the point, because it is linked to the first slide I showed, the bullet point which says that in terms of this elicitation from the AABB's Uniform Donor History Questionnaire, which is the standard way of selecting blood donors in the United States, the rationale for answers was framed as much by the questionnaires' general purpose in the mind of the donor as by the specific topic of individual questions. What does this mean? This means that most donors basically self-assess themselves relative to what they think is the intent of the selection procedure, i.e. the intent of the selection procedure is to decide, is my blood safe or is not my blood safe? They make a self-based decision on this fact, irrespective of whether they are in an MSM or non-MSM, compliant or non-compliant category. In other words, there are numbers of blood donors who, again, according to the letter of the questionnaires, the letter of the selection procedures, the letter, the detailed letter, if you like, of the questions, disregard this and hone down, rightly or wrongly, to the intent, which is my blood safe? And this is the reason why many of the current publications show, as I showed in the first slide, that a considerable number of individuals are actually not compliant if you look at the letter of the law. Next slide. However, this is all by the by. I think that it is important to note that many of the considerations which I outlined 2 years ago were based upon modeling type exercises in regards to the whole question of the risk. And if you look at this paper here, which I believe is from Canada, like many of the excellent publications in this field, you will see that the modeling actually showed that the risk would go up by a certain amount, but the actual observed HIV positivity in the donors who were allowed back into the donor pool is considerably lower. Now, this is a subject which demands much more reflection and assessment than I have time to do today, but it does show you that in the end you have to have active surveillance mechanisms and the data generation rather than rely on hypothetical models based upon what I might call somewhat dubious epidemiological presumptions. The next slide, please. I will now say something, because it was mentioned in my invitation, on the question of hepatitis E virus. And there's much more on this in the expanded set, and I refer you to many excellent reviews on this agent, its epidemiology, modes of transmission, and so on. This is just the global distribution. Next slide, please. Thank you very much. This now starts to hone in on the plasma industry. This is a survey from the commercial plasma sector showing the actual positivity in plasma donors in the United States, which are, of course, key to the safety and the supply of the blood of the plasma system worldwide. You can see that basically the observed prevalence is extremely low in the plasma donors. in the United States. Next slide, please. And this is much more important because, as I and many others have reiterated over many, many years, really all these issues about selection and testing are secondary, if not irrelevant, to the actual safety of products. What's important is the capacity of the systematically introduced viral inactivation steps in the manufacture, and without belaboring too much the point and going into too much detail, here you've got a selection of data on different types of viral inactivation procedures. There's nothing here in regards to factor VIII, but there is available information which basically shows that in terms of hepatitis E itself, culturable and able to be analyzed directly, and in terms of what is agreed by experts in the field, which by no means include myself, in relation to model viruses, you can see that the various processes are very effective in inactivating the agent. Next slide, please. Now, I thought I would share this slide because this is one of these promiscuous slides which everybody has basically had, but this is from a recent review, and it's actually very interestingly worded, this review, because it's basically a gentle hint to the current United States administration to not fool about too much with the budget of the CDC. But one of the main authors of this interesting review, and I commend it to your attention, is Dr. Fauci, which I think he's about to retire. And he makes the point that in 1984, he started a map and he put on it the one emerging agent, which was HIV. In 2017, his map looks like what's on the right-hand side. In other words, thanks to the ceaseless manipulation of the environment, our continuous destruction of nature, as well, of course, as modern practices of travel and so on, emerging agents continue to emerge and re-emerge and are very quickly spread. So there's no room for complacence in relation to this issue as far as it goes in terms of blood safety. Next slide, please. But we have much room to be reasonably cautiously comforted by the data from the industry. I don't want people to think I'm advocating here one particular company. It is just that this set of data does include a spread across the three main product groups, but you can see that in this instance, Zika is very well inactivated by the tried and tested, and in my opinion, should be mandated method wherever it can be, solvent detergent treatment. This is data from Octapharma. The next slide is another set of data from CSL, purely and simply to show that the same situation occurs for a range of these viruses, as you can see over there, including SARS, West Nile, Zika, and so on, in terms of pasteurization. So the processes we have work for what we know and what we don't know, but what seems to come out as a result of the changes in environment which I outlined before. The next slide, please. I now want to say something, and I know that this was not mentioned in the letter, but I feel that this was an event important enough to make this group aware of it, and this was the somewhat disturbing report which came from the Edinburgh group, Professor Hill and his colleagues, in regards to the possibility of sporadic CJD. I emphasize we're talking sporadic here, in 2 plasma product recipients in the UK. Many of you are familiar with the report, and I encourage you to look at the publication. It is a very, very well-worded publication seeking to share the knowledge without imparting any level of alarm. Next slide, please. The reason why I'm putting this up is to remind you that in terms of prionic diseases, and we have known this for many, many years, although the focus of attention in terms of human transmission through blood has been on variant CJD because of undoubted causal proof, we have always known through animal models that transmission can occur through the blood also in other forms of prionic disease, including the sporadic form. The reason why this has happened is something which requires many days of discussion. But this is a slide which is a summary from many studies, many of them from Bob Brower's group, which shows that this is indeed the case in many animal models. Next slide, please. Now, in the past year, we have seen 2 key publications which actually have managed to, using technology which, if there was the will and the money, could be easily adaptable to blood bank screening, can actually detect VCJD and other prionic forms in blood in a way which is amenable to mass screening. This is just one of them. This is from a French group, I have here shown you the abstract, and I'll show you data from the publication in a second, which shows that the assay allowed the detection of silent carriage before the clinical onset. In other words, in what we could call, if you want to be very liberal with your language, the window period in 2 blood donors who subsequently developed VCJD. So this is of great interest to the blood community. Let me ask you for the next slide, please. And this is the principle of the assay. It harnesses the well-known principle of the affinity of prion scrapie and similar dysfunctional forms of the prion protein for plasminogen in a capture-based mode. You can see it up there. And there are some readouts from the assay which are based upon blotting technology. You can also get readout with this assay if you want, using a fairly standard colorimetric methodology. Now, the point I want to make to you is that in this assay, there was a very nice demonstration of sensitivity to VCJD, but there was also one sample which was unquestionably a sporadic CJD patient on the basis of the clinical data, which turned up positive in this assay, which I think shows that in this system, in this study, there was infectivity in the blood of sporadic CJD patients. Next slide, please. This is from another paper which is reasonably recent, which shows blood infectivity in variant and sporadic CJD using intracerebral inoculation. Now, of course, we all know, those of us who've had to study this disease over these many years, that injecting stuff in the brain is a considerably different kettle of fish to actual intravenous infusion. And in fact, there is data to show that the relevant incubation periods are, of course, extremely different and affected by the route of infection. But it is the case that, again, in this demonstration, you can see that there is infectivity in the blood in all forms of pathologic prion. And I seek to make this point without wanting to get people too upset and emotional, that a prion is a prion is a prion. And in my opinion, the differential infectivity in variant CJD relative to the sporadic form has always been attributable not to the infectivity per se, but to the different distribution of the evolution of the prion in the body. In other words, a very higher level of infectivity in lymphoreticular tissue in VCJD, which then results, of course, in a higher titer in the blood. But it is a question of titer, and in relation to titer, then that translates basically into a question of incubation period. Next slide, please. This is an interesting slide because in one of my major roles currently, I work in Italy, where the Italian regulatory authority, acting upon the advice of the equivalent of the CDC in Italy, the Istituto Superiore di Sanità, an immensely credible and able organization full of very good scientists, has always maintained that in relation to blood and plasma pools, demonstration of a sporadic CJD donors demands, as far as their market is concerned, withdrawal. Now, this is based upon the work of Massimo Pocchiari, a friend of mine. We have served on various committees over many years, who has a large body of both epidemiological and experimental data. The experimental is pretty much similar to what I've already shown you. In fact, some of those studies come from his group, which show that this is indeed the case. And this is a paper published some time, which shows that there is an association based upon his multivariate analysis between a long incubation post-blood transfusion observation and sporadic CJD. In other words, people with sporadic, who get blood from sporadic CJD donors, if you follow them long enough, there is a significant association. It comes back to what I've already said in terms of incubation period. Now, we don't have the time, although I'd be very happy to discuss, to analyze this in detail, but you have to remember that the epidemiology of blood transfusion itself is centered mostly upon people who are actually old, and in all probability, if they do cop a sporadic event, they will die before the CJD. And in fact, this is also the case for variant CJD, which is what happened with the famous case of the hemophilia patient, which I will allude very briefly, I think, in my last slide. Next slide, please. Okay, now to go back to our comfort in relation to plasma derivatives, this is just one of many, many studies amongst many people, synthesized particularly by the Pathogen Safety Steering Committee of the PPTA, chaired by Dr. Kreil, who obviously you all know, which studies the distribution of prion. And in many of these studies, the actual prionic strains were actually sporadic strains rather than variant strains. This is something which many people don't actually fully emphasize because of the cognizance that a prion is a prion is a prion. And you can see that the distribution of the prion is fortuitously, this is, you know, basically the spirit of Edwin Cohn guiding and benefiting us away from the therapeutic fractions of interest, except for factor 8. Next slide. And so for factor 8, we need more. And this is an interesting demonstration of the contrast elicited by having different levels of clearance in actual patient-related type incidents. On the left, you can see the publication related to the famous case which came about in 2008-2009, reported by James Ironside and colleagues in hemophilia, in which, as you will remember, a patient who was totally asymptomatic for VCJD died from unrelated causes, and since he was part of the group of scrutinized patients in the Edinburgh cohort of people who have definitely received products from infected donors, was well studied and autopsied, and as you will remember, a single sample from histologically examined spleen was found to be positive. I reiterate, the patient was not sick from VCJD, and there was no evidence of disease otherwise. The other, I apologize, as usual, I try to cram 2 slides into one, is, to my mind, a very interesting paper which was published pretty soon after the British hemophilia case, but has been reiterated recently in an interesting survey from the UK, in which a lady who received IVIG from a similarly contaminated, quote-unquote, UK plasma pool, in other words, again, this was like the patient with hemophilia, she definitely received product which was made from VCJD donors, and died, was examined from unrelated causes, died from unrelated causes, was examined, was assessed, and there was absolutely no indication of infection. Now, of course, I think that we would agree that the probable main reason for this is the different level of clearance in IVIG manufacture, which is huge and definitely removing much of the infectivity, compared to what we know was the product mostly exposed by the patient with hemophilia, which was the British product, otherwise known as 8Y, which has a very low level of clearance. This is well described in the paper, as well as in other documents published by the government of Britain at the time. So I think I'm approaching the end of my talk. Can I have the next slide? So I would say, and I hope that I have carried out my charge, I hope to the chairs, that the deferral relaxation for MSM has possibly increased HIV risk only to the transfusion, hypothetically to the transfusion of fresh components, because of mostly the fact that we don't have any problem with absolutely obliterating HIV from all plasma products today. And this has been heightened. The first part of this bullet point is what I stated basically 2 years ago, but I think that this has been heightened by the introduction of pre-exposure prophylaxis for the reasons that I have outlined in the first part of the talk. I would reiterate that modeled predictions which indicate that there is a higher risk to blood safety should be taken seriously. However, modeling has to be buttressed and followed up by actual active surveillance. And active surveillance so far has not indicated that this is the case. Emerging, re-emerging, and re-emerging, we now have re-emerging agents, continue to threaten the blood supply. There is no doubt that they are a threat for transfusion of components. And this is why it is a matter of urgency for the fresh blood sector to follow the example of the plasma industry. And now that the technology is definitely available to introduce viral inactivation for fresh blood components, this is coming across in several countries, but it is not yet a universal requirement, and the main reason is basically the expense.
And in relation to my comments about prionic disease, I would say that we do have findings indicating that pathogenic prion strains other than the variant form are also transmissible by blood. Current clear levels are high enough to give comfort, if not complacency. And I round off by, I hope, absolving myself from all accusations of alarm by assuring you that plasma protein therapies manufactured under GMP and under adequate regulatory oversight are safe and effective medicines. Thank you for the attention.